This is Mr. Wiersma. This presentation is on DNA and how the structure of DNA was discovered and some of the people involved in figuring out the structure and shape of DNA. I'd like to start off with a guy named Griffith. He was in the 1920s and he was working with a bacterium called Streptococcus pneumoniae. And some strains of this bacteria are virulent. That means they cause pneumonia and make the mouse or host organism sick, and other ones are not virulent and they don't make the organism sick. The virulent kind would be the smooth strain. Those have a smooth capsule around the bacterium, and the ones that are not virulent are the rough strain. So I'll just give you a quick summary of what he did. He took, as you look at the picture, he took some of the bacteria, the rough strain, again, not virulent, and he injected those into the mice, and as you would expect, they did not die. Then he injected some of the smooth strain bacteria. Again, those are the virulent ones, the ones that cause the pneumonia disease. He injected those in the mice, and as expected, they died. Then he used heat to kill the smooth strain. And again, as you would expect, because he killed the smooth strain bacteria, they did not kill the mice. But then what he did is he took the heat-killed smooth strain, which again are virulent, and he mixed them with the rough strain, not virulent bacteria. And to his surprise, when he took that mixture and injected it in the mice died. So he concluded that somehow, even though the smooth strain bacteria had been killed by heat, they were giving their information to the rough strain bacteria, and he called this transformation. Again, DNA is all about information. The next people I'd like to talk about are Hershey and Chase. Hershey and Chase worked with bacteriophages. These are viruses that infect bacteria. And what Hershey and Chase wanted to find out, what part of the phage, the virus, was actually attacking the bacteria, infecting the bacteria. So what they did was kind of smart. They labeled the protein coat with a radioactive isotope called sulfur 35, and they labeled the DNA with radioactive phosphorus 32. And then they mixed these phages, these viruses, with the host bacteria. And at the conclusion of the experiment, they found that just the phosphorus 32 was inside the bacteria. So this showed that the protein coat generally stays outside in the case of the phage, where the DNA goes inside the bacteria. And so this showed that DNA is the genetic material and not proteins. The next people I'd like to talk about are Wilkins and Franklin, and I believe they were both from England. And if you look, that's Rosalind Franklin on the left, and the picture on the right is called P51. That's the famous X-ray picture of DNA. And they did their work, and unfortunately, shortly after this photo was published, Rosalind Franklin got cancer and passed away. But Wilkins and Franklin were key to discovering the shape of DNA, because later on, as we'll learn, two gentlemen named Watson and Crick used this photo 51, to deduce the double helix shape of DNA. The last people I would like to talk about are Watson and Crick. Again, they did their work in the early 1950s, and they are the ones that are given credit for determining the double helix structure of DNA and how the bases pair with each other. And if you look at the diagram to the left, you'll notice that it does look like kind of like a twisted ladder, and the steps of the ladder are composed of these things called bases. The bases are guanine and cytosine, and adenine and thymine. And the sides of the ladder are made of repeated units of phosphate and sugar. And also notice that guanine always pairs with cytosine, and adenine always pairs with thymine. This last slide is showing you the three-dimensional shape of DNA, or deoxyribonucleic acid. And notice that it does look like a twisted ladder, with the steps of the ladder being made of two nitrogen bases. Again, adenine pairs of thymine and cytosine pairs of guanine. And the vertical parts of the ladder are made of repeating units of sugar and phosphate. Again, this is a beautiful molecule. This is the molecule that stores your hereditary information, and the structure was not determined really until the 1950s.